is the solubility standard for peptides defined?
Hello. Different peptide products have different dissolution conditions and need to be judged based on specific sequences.
How to make a judgment?
Peptides are generally composed of 20 natural amino acids, and it is necessary to clarify the relative properties of the 20 amino acids. According to the combination, it can be roughly divided into hydrophilic sequences, hydrophobic sequences, acidic sequences, and alkaline sequences.
Today, I would like to introduce to you the 4 suggestions for excessive dissolution of the above sequences. What are the hydrophilic sequences included? When hydrophilic amino acids are evenly distributed throughout the entire sequence, and the sequence contains greater than 25% charged amino acids and 25% hydrophobic amino acids, it can be considered a hydrophilic sequence. We generally recommend dissolving it in sterile distilled water, deionized water, or buffer solution. What are hydrophobic sequences? The number of hydrophobic amino acid residues is greater than 50%. Even if the sequence contains hydrophilic amino acids, the polypeptide may not be soluble, or only slightly soluble in water. Suggest adding ultrasound-assisted dissolution and cosolvent, such as DMSO, DMF, acetic acid, acetonitrile, methanol, propanol, or isopropanol. Suggest dissolving the peptide into a high-concentration solution first, and then slowly add water or other buffer solutions. What are the acidic sequences included? The residues of glutamic acid and aspartic acid are greater than those of arginine, lysine, and histidine. It belongs to acidic peptides. If pure water cannot dissolve it, please try using PBS or 0.1 mol ammonium bicarbonate solution to dissolve it and dilute it to the desired concentration. What are the alkaline sequences included? The residues of arginine, lysine, and histidine are greater than those of glutamic acid and aspartic acid. It belongs to alkaline peptides. If pure water cannot dissolve it, please try using 10% minus 30% acetic acid. If the peptide is still insoluble, dissolve it in pure acetic acid or trifluorosilicic acid TFA, and then dilute the peptide solution to the desired concentration. Are there any other special circumstances? If the peptide sequence contains cysteine cis and requires liquid storage, it can be dissolved using dimethylformid or unmethylpyrrolidone. Do you have any suggestions for cell experiments? If peptides are used in cell-related experiments, the concentration of DMSO should not be too high. Otherwise, it will have toxic effects on cells. For DMSO concentration control, a concentration below 0.1% is almost safe for all cells. Although there have been cases where 5% DMSO has been successfully applied to certain cells, we recommend not exceeding 0.5%. If you have any questions during the experiment, you can contact our online customer service, and we will be dedicated to serving you.